Namaste everyone, I'm your Dr. Ramesh Kasarla. In this video, we will delve into the detailed exploration of the human reproductive system. Before proceeding this one, I encourage you to watch previous videos on the male reproductive system and spermatogenesis for the better understanding. The male reproductive system and spermatogenesis integral to the sperm production, storage, transportation as well as release of the semen and urination. Spermatogenesis is the process which the sperm cells productions occurs. The germ cells gives rise into the haploid spermatozoa. This sperm productions takes a place inside the semi-inferior tubules, which is a convoluted clusters of the tubules located inside the testes. Normally, spermatogenesis process begins before the birth to formation of the primordial germ cells during the embryonic development. These cells are derived from the primitive streak during the early embryogenesis, migrate into the genital ridges and eventually settle in the developing testes. These cells are enter into the testes. These primordial germ cells, after settling into the testes, differentiated into the spermatogonia, also called as the stem cells for the spermatogenesis. This spermatogonia remains quiescent until the puberty. It will take 12 to 14 years. Spermatogonia cells are undifferentiated diploid cells responsible for the sperm production through the spermatogenesis. During the puberty, hormonal changes stimulate the spermatogenesis. The hypothalamus releases the gonadotropin releasing hormone and promoting the anterior pituitary gland to secrete the follicle stimulating hormone and also luteinizing hormone initiating the sperm production. Spermatogonia cells are undergo to the mitotic division to maintain the constant fold for the germ cells. This mitosis, it is the type of the cell division that occurs to the somatic cells for the growth, repair and sexual reproduction. It's involved in the several distinct stages. Here we can see the different stages of the mitosis. First one is the interphase. During this phase, cells are grown and DNA replication will take place. Second one is the prophase. During the prophase, chromatin condenses into the chromosomes and nuclear envelope breakdown. Third one is the metaphase. During this phase, chromosomes align at the metaphase plate. Fourth phase is the anaphase. During this phase, sister chromatids are separated and move into the opposite folds. And the next one is the telophase. During this phase, chromosomes are decondensed and nuclear envelopes reform. Final step is the cytokinesis. During this phase, cytoplasm divides and forming two daughter cells. Some spermatogonia differentiated into the primary spermatocytes, initiating the meiosis, which is the process for the next leading to the sperm production. This is ensure the continuous supply for the sperm cells and reproductive functions. Now, primary spermatocytes are diploid cells that rise from the differentiation of the spermatogonia within the semi-inferior tubules of the testis. Upon this differentiation, primary spermatocytes undergo to the DNA replications, resulting in the duplication of the chromosomes. This replication process ensures that each primary spermatocytes process the two set of the chromosomes, maintaining the diploid state in the full complement genetic material, means 46 chromosomes. Now, primary spermatocytes are enter into the meiosis process. During this meiosis, it is a type of the cell division that occurs in the germ cells, leading to the formation of the gametes as a sperms and egg cells. It's involved into the two consecutive divisions of the meiosis, resulting for the four haploid daughter cells. Here the diploid primary cells are going to change into the haploid cells. Here we can see the step-by-step process of the meiosis. First one is the prophase. During this prophase, chromosomes are condensed and homologous chromosomes are pair up and crossing over the occurs. It is leading to the genetic recombination. Next one is the metaphase. During this phase, paired homogeneous chromosomes are aligned at the metaphase plate with the spindle fibers attaching to the each chromosome centromeres. Anaphase. During this anaphase, homologous chromosomes are separate and move towards to the opposite poles of the cells, pulled by the spindle fibers. Final one is the telophase. During this telophase, chromosomes are reach the poles and the cell undergo to the cytokinesis, resulting to the two haploid daughter cell formation. During this spermatogenesis, meiosis 1 is a vital stage where the homologous chromosomes are pair up and exchange the genetic material and leading to the genetic recombination and formation of the diverse haploid secondary spermatocytes. Secondary spermatocytes are formed during this process. Our haploid cells are vital for generating the genetically derived sperm cells capable of the fertilization. These cells are having the half of the number of chromosomes as the parent cells and contributing to the genetic variation among the sperm cells. Next one is the meiosis 2. So second stage of the meiosis. It is similar to the mitosis, makes a second division of the meiosis. In this phase, secondary spermatocytes resulting to the form the meiosis 1 undergo to the further division to the yield of the haploid spermatocytes. This process involved in the second stage of the meiosis. Here we can see the different stages of the meiosis 2. Prophase 2. Each haploid daughter cells undergo to the brief prophase 2 where new spindle fibers form. Prophase is followed by the metaphase. During this metaphase 2, chromosomes are aligned at the metaphase plate in each daughter cells. And next phase is the anaphase. The sister chromatids are separated and move towards the opposite poles of the cells. Final one is the telophase. During this phase, chromosomes reach to the poles and cytokinesis occurs again. During this cytokinesis process, that cytoplasm will divide and they will form the new cells, resulting in 4 haploid daughter cells formation. Each is having the half number of the chromosomes as the parent cells. So as previously I told, diploid cells are now changed into the haploid cells, means 46 chromosomes are now having the 23 chromosomes as a haploid cells. So sperm cells and egg cells are having the 23 chromosomes. Subsequently, these haploid spermatid immature cells undergo to the morphological transformation during the spermatogenesis to develop into the mature spermatozoa. For formation of the mature sperm cells, it will enter into the differentiation. These spermatids undergo to the extensive morphological changes and differentiation into the form of mature sperm cells, also called as the spermatozoa. This includes the condensation of the chromatin in the nucleus and formation of the aerosomes and the development of the flagellum. These mature sperms are fully mature spermatozoa and released into the lumen of the semi-inferior tubules, and then it moves into the epididymis for further maturation and storage. They are eventually ejaculated during the sexual intercourse and capable of the fertilizing the egg. This is the complete step-by-step process of the spermatogenesis. This is the entire process takes place approximately 64 to 72 days to complete, to form the initial differentiation of the spermatogonia to release the mature spermatozoa. It is highly regulated in the coordinated process essential for the male fertility and reproduction. Until now, we get to know about human reproduction system and individual parts and functions and spermatogenesis, and which are the main functions involved in the mitosis and meiosis. In our upcoming video, we will delving deep into the intricate individual structures and functions of the sperm cells. For more details, be sure check out my channel Dr. Ramesh Kasarla. Feel free to leave your suggestion in comment section below. Thank you. Dhanyavad. Be healthy.